Before we begin, let's define some terminology. Meiosis is a division of a diploid nucleus in order to produce 4 haploid daughter cells for potential use in fertilization. Before meiosis can begin, the DNA in the cellular nucleus must form itself into multiple sets of homologous chromosomes. Homologous chromosomes contain identical or almost identical, as we shall later see, information to each other. N is the number of sets of homologous chromosomes in a given cell. The value of n is constant in all the cells of a given organism. What is the value of n in this particular cell? Each dot represents a particular set, and the chromosomes which have the same colored dots are homologous. In this case, we have 3 different dot colors: red, yellow, and green. Therefore, the value of n is 3. Ploidy is the number of chromosomes in each set. What is the ploidy of this cell? Each colored dot appears around 2 different chromosomes. Therefore, this cell is diploid. Once this can begin, the chromosomes have to replicate. The number of chromosomes can be determined by counting the number of centromeres, which are being represented by black dots in between the 2 sister chromatids. There are 6 centromeres. Therefore, this cell has 6 individual chromosomes. What is the new value of n? Since the number of chromosomes didn't change, neither did the value of n. There are 3 sets of chromosomes, and therefore the value of n is 3. What's the ploidy of the cell? Well, both the value of n and the number of chromosomes haven't changed. Therefore, the cell remains diploid. In prophase 1, homologous chromosomes pair together. This is done by comparing the chromosome's DNA sequence until a matching pair is found. The sequences are compared through the process of crossing over, which involves the comparison and exchange of corresponding segments between 2 homologous chromosomes. However, not all homologous chromosomes are identical. For example, humans share about 99.5% of their DNA, but there is still that 0.5% difference in each individual's genome. Sometimes, when crossing over, the segments in change contain that difference, resulting in gene recombination. In metaphase 1, the chromosomes are lined up along the center of the cell. Centrosomes use spindle fibers to tug on one of the 2 homologous chromosomes, eventually causing them to end up in the center. Anaphase 1 is the separation of the homologous pairs. Each centrosome pulls its chromosome to its end of the cell, separating the pair. However, the sister chromatids remain intact. In telophase 1, the chromosomes are separated into 2 individual cells. We are now in meiosis 2. How many chromosomes do we have now? Once again, we can determine this by counting the centromeres. This cell contains 3 centromeres and therefore 3 chromosomes. The same can be said for the other cell. What is the new value of n? The value of n is still 3, since each of the 3 chromosomes contain different information, making them each a set of 1. This answers the next question: what is the ploidy of this cell? Since each set contains 1 chromosome, the cell is now considered to be haploid. Metaphase 2 is similar to metaphase 1. The centrosomes once again use spindle fibers to line up the chromosomes in the center of the cell. However, in this case, it is the sister chromatids that are lined up in the center of the cell in order to be separated. In anaphase 2, the centrosomes break apart the centromeres and separate the sister chromatids. In telophase 2, the cell once again divides, giving us 4 unique daughter cells. So, how many chromosomes do we have now? Each cell contains 3 unique chromosomes. Since each of the chromosomes are unique, the value of n is also 3. And the ploidy of the cells? 3 unique chromosomes without a pair make each cell haploid. A quick note on X and Y chromosomes. Although they are different, they still contain an identical sequence in the pseudoautosomal region at their tips, allowing them to undergo crossing over.